Hi students, in this video we will be discussing about the procedure involved in plant tissue culture or the techniques which is involved in plant tissue culture. Plant tissue culture technique, we can classify it into major steps, three major steps. The first step is we have to prepare. We have to take certain preparations, and so we can call it as the preparation stage. And here we have to prepare the nutrient medium, we have to select the explant and have to sterilize the explant, and we have to inoculate the explant. In the second stage, that involves the subculturing process, and here we do the subculturing. That is nothing but the transfer of tissues which are regenerated to a fresh medium, and there they can again undergo cell proliferation and callus formation. And the third step or the final step is the transfer to an oxygen-rich medium to induce the roots. When the plant develops both shoot and root, then we transfer it into the glass house or the greenhouse for hardening process. So these are the three major steps. First step is the preparation stage. Second step, where we subculture the tissues or the shoots, and then involves the acclimatization, rooting and acclimatization stage. Okay, so the first step, that is the explant selection, it is the first step, and most responding or most actively growing tissues are taken for culturing. So we select the most actively growing tissues as the explant, and it should be free from any disease also. And there should be the presence of parenchyma cells in the explant, which is marked as an important condition for selection of explant. Okay, it's because parenchyma cells, they are highly responsive, they quickly respond in the culture medium. These explants, after selection, they are cut into small pieces approximately 2 to 5 millimeter or up to a maximum of 1 centimeter in size. Then this second step involves the explant sterilization. Okay, so the explants, they must be sterilized to remove the microbial contaminants, if any, present in that explant. So in this step, we treat these explants in with different detergents, fungicides, or any other sterilants, and finally we sterilize it with mercuric chloride, or in some cases we may sterilize it with sodium hypochlorite also. Okay, so the second sub step is the sterilization of explant. Now another preparatory step we need is the preparation of nutrient media. We have already discussed the preparation of tissue culture media in our previous video, so that all steps I don't need to explain it again. So we have to prepare the nutrient medium, and then the next we have to we have to inoculate these explants. Okay, so it is nothing but the inoculation is nothing but the process of transfer of these sterilized explants to the nutrient medium in aseptic conditions. So this inoculation process, it is done in aseptic conditions. So for that we use the laminar airflow chamber. We do the inoculation process in laminar airflow chambers, and these culture vials, after inoculation, they are kept in the culture room or they are incubated. The culture room is having controlled conditions. It will be having 25 to 28 degrees Celsius temperature, and the photo period is also maintained, and also the relative humidity is also under control. So these inoculated culture vials are kept in the culture rooms for incubation. So that was the step. And after that, the second step comes. The second step is the subculturing. Subculturing, when the callus is grown or the shoot is initiated, it is we have to transfer the tissue to another fresh medium after a period of time. Say for after 4 weeks or after 3 weeks, we have to transfer the tissues, the initiated tissues, or it may be the callus, to a new medium or the fresh medium. Otherwise the nutrient will be depleted in that culture vials. So what happens is toxic metabolites accumulate in that culture vial, and that will be toxic to the plant. Okay, so for that, and it may lead ultimately to the death of the tissue. So for that, we have to transfer the tissues continuously within a period of 4 weeks to a new or fresh culture vial. So that step is called subculturing. So the multiplication, in the multiplication step, the explant give rise to the callus, or it may be a shoot initiation. So which is manipulated by varying sugar concentration and the auxin cytokinin ratios to form multiple shoots. The callus may be subdivided a number of times. So according to the ratio of the auxin and cytokinin, it is induced to form multiple shoots, or it is induced to form the callus. And next step is root formation. In root formation, the shoots are transferred to a root-inducing medium where the auxin-cytokinin ratio will be higher, which induces to form the roots. So we have already developed the shoots. That shoots are transferred to subcultured to fresh mediums, and after reaching an appropriate size, these shoots are transferred or subcultured to root-inducing mediums, which will be having a higher auxin to cytokinin ratios. So in that medium, root inducing medium, it will develop the roots. So that is the next step. And after the roots are developed, the rooted plantlets are then taken for hardening process. They are deflasked and they are transferred to a potting mixture, and here they are acclimatized, or this is the process of hardening process. We transfer it step by step to the ex vitro conditions. So after taking it to the greenhouse, we then gradually take it out to the field conditions or to the outside conditions. So these are the steps which are involved in the tissue culture process. So first, in brief, there are different steps. First step is preparatory. Here it involves preparation of nutrient medium, explant selection, explant sterilization, and explant inoculation. After inoculation, it develops new shoots if it is a shoot inducing medium, or if it may develop a callus. So both may be the condition. In both the cases, they are taken to a next or fresh medium after a 4 weeks period, and that process is called subculturing. Subculturing is essential because allowing it to remain in that same culture vial, it may cause depletion of nutrients after a period of time. The nutrients will be depleted, and also it may lead to accumulation of metabolites which may be toxic to the plant, and that ultimately leads to the death of the tissues. So we have to transfer it to a fresh medium in a regular interval of time. So that process is called subculturing. After subculturing, when the shoot attains an appropriate size, it will be transferred or subcultured to a root inducing medium where it induces the root, and the rooted plantlets are taken for hardening process, and they are transferred to pots, sterilized potting mixtures, and then taken out step by step to the greenhouse and to the field. and finally the hardened plants are obtained. These are the steps involved in tissue culture.